studies of the summer school. Today's theme is biogeochemical processes, and I have the pleasure to introduce you today's morning speaker, so I hope you're going to Audrey Sawyer, if I pronounce it correctly.
Yeah, you did great.
So the floor is yours.
Thank you. Well, I want to start by just saying thank you so much to the organizers of this beautiful 2 weeks to be able to interact with hydrogeologists, hydrologists, engineers, chemists, people studying all kinds of cool things involving mixing. And so today, I'm going to take a look at an area where mixing is really important that I've spent most of my career studying. And this talk is really the compilation of a team effort, including current and past members of my lab group at Ohio State University. And I'll also highlight some work that I'm a part of on coastal hydrogeology with the Universitat Politècnica de Catalunya. Where are we going to go with this talk? I'm first going to talk about why I care and why I hope you'll care about surface water-groundwater exchange and mixing processes in aquatic settings. We'll first spend the first half of the talk looking at interfaces in rivers and introduce the concept of the hypereic zone. I'll talk a little bit about how our group and others have used some continuum scale models to get at these flows and exchange processes. And we'll talk about some of the metrics that we care about for thinking about biogeochemical and thermal implications in these zones. And I'll give you a little bit of an example from some of our work in a high mountain stream. But then I'm going to whisk you away from the mountains to the coast, and we'll talk about something very relevant to what's right outside of our door, coastal interfaces, and the subterranean estuary. So we're going to add salt water to some of these things that I will have talked about in the previous talk. I'll take you on a virtual field trip right outside of our door, and I'll just talk a little bit about some frontiers in this space. Why care about fluid exchange across a seabed or a riverbed? It turns out it's important for all kinds of processes, including diagenesis and preservation in the rock record. It's not all just molecular diffusion as things are being deposited and rocks are being created. And then, and I don't need to say too much more about that in this community since we've been focused so much on transport this week. Elemental fluxes to rivers and oceans are controlled by the transformations that happen at this interface. It's important for benthic ecology and something that our group has spent some time thinking about, contaminant processing. So when you, for example, get a pollutant in a stream, how long is it going to stick around, due in part to that residence time in immobile zones like the stream bed sediments. Let me introduce to you the hypereic zone, which is the region beneath and alongside of river channels or streams where there's mixing of shallow groundwater and surface water. And mixing happens at a variety of scales with a variety of rates. And this zone is very important for all kinds of things, including it's where fish sometimes lay their eggs. So it has ecological significance. It shapes things like the community of macroinvertebrates that are in streams. And so it has ecological relevance as well as relevance to thinking about contaminant transport and nutrient cycling in streams. And Camille, I believe it was, started us off thinking about Toth type of flow through landscapes a little over a week ago now. I like to think of the hypereic zone as essentially like a mini Tothian flow problem, where you have some scales of local mixing or local flow that goes through smaller roughness elements in the riverbed, and then you have deeper flows that connect up multiple different features in a riverbed, and what's essentially regional flow in the Toth system. In this case, it's flow that connects up different parts of rivers over the reach scale, say. And each of these has different residence times and affects different chemical processes along flow paths. We can represent this flow that is essentially like self-similar in nature with thinking about the bed form as a representative feature. When you look at streams, they have different features that contribute to roughness, but one common one, especially in sandy beds, but also in gravelly beds or muddy beds, are bed forms. And they have, you know, basically ripples or dunes. If you're a sedimentologist, you spend time classifying these things and how they form and how they move, but they're all self-similar. And so we can use the bed form as a representative element for thinking about solute, fluid flow and solute transport through the bed. And if you have a train of these things, basically what happens is there's flow of surface water over the bed form crest and trough. And we get a zone of high pressure as surface water is moving against the bed form face. And then in the recirculation zone and right at the crest, we get a zone of lower pressure. And all of that drives downwelling through the upstream side of the bed form and upwelling downstream. And because all of this exists in a stream that has some general slope to it going downstream, we also have this underflow that's always kind of trucking along down below and not necessarily interacting with the bed unless you have some sort of overall hydraulic head gradient that's driving upward or downward flow on top of these processes associated with bed form current interactions. And we can idealize this because running a turbulent CFD model coupled to traditional groundwater flow model is computationally intensive, but we can just use some basic tricks. It turns out that the component of pressure that drives flow along the bed can be represented just more or less as a sinusoid. And so if you picture, let's see here, if you picture that the bed form is lying something like along this yellow line, that's the sediment-water interface here, the high pressure zone is sort of shifted by a quarter cycle upstream of that. And the low pressure zone, obviously 180 degrees downstream of that. And we can represent flow through the bed in response to just this simple idealized sinusoidal hydraulic head signal along the surface. And that becomes really tractable for doing all kinds of analytical solutions and simpler, larger scale models as well. And we can start to think about things like the residence time distribution of solutes as they enter this sediment bed here. And it turns out, like many things we've been talking about this week, that the residence time distributions are heavy-tailed. And so what that means is that most of the solute is going to flush really quickly from the shallow part of the bed, but there is going to be, this long tailing of solute, this persistence due to some of these deeper, slower flow paths and mixing processes that happen beneath these bed forms. And so this is all a very convenient way of thinking about streams, and it sits within the broader concept of the fact that bed forms contribute to some amount of, they're an immobile storage zone. They contribute to solutes sticking around in the stream bed for some amount of time with their own sort of residence time distribution. But then you have larger elements. So this is thinking about a meandering river, the flow through the floodplain from one side of the stream to the other. And there's some residence time distribution associated with that. And then, of course, thinking about even longer flow paths, starting to approach Tothian type of scales. And what we see is that at the end of the day, the hyperrheic zone is just one part of this whole self-similar transport behavior within catchments. The bed form is also really convenient for doing some models at the reach scale, and this has been a fun area for our group here. I'm showing you some of the original work by Susa Stonedal, looking at ways to take a channel and all of its bed forms and basically just do some conformal mapping to say, what are the different wavelengths of these bed forms for any complex topography in the stream bed. You can just decompose it into different wavelengths, and because groundwater flow follows the law of superposition, you can just put all of these different head gradients together and solve for fluid flow through the bed due to a variety of different features. You know, the full complex distribution of bed form topography, or just looking at, for example, one wavelength. And you can start to think about, when solute sticks around in a riverbed, what are the features in the riverbed that are driving that? It's also a convenient, this use of the bed form as a building block is also a convenient method for upscaling. And so I want to take a little bit of time in this talk to highlight some ways that we use this information to upscale, and partly through the work of Jesus Gomez Velez. So this is not our group's work, but I'm an admirer. So I'm going to show you a little bit about how this concept of thinking about streams as basically compositions of bed forms and meanders can be used to understand how water cycles through the stream bed as it's headed downstream. And so this map shows you a large chunk of the Mississippi River Basin. And what you're looking at here is the number of hyporheic excursions per kilometer. So if you were to drop a little bit of a contaminant that doesn't want to break down into a stream and ask yourself, how long is it going to take to move through all of these mobile and immobile zones as it moves downstream? How many skips is it going to take through the hyporheic zone? We can actually map that by thinking about what the permeability is that we would expect in different parts of the river reach, and what the flow depths are that we would expect, and what some of the wavelengths of bed forms are that we would expect. And you get really cool maps like this that show that in the larger main stems of the river, you're going to make a lot fewer excursions through the hyporheic zone per kilometer traveled downstream. So it's the tiny little streams that are most efficient at cycling solutes through the hyporheic zone. And so this raises the question, if we want to do some upscaling for any model, what are the metrics that we care about? And I've already talked a little bit about fluid exchange, fluxes, and residence times. But we also want to think about how those two relate to drive the degree of chemical transformation or processing within the stream bed. And the reason is because the stream bed is, it's a really reactive place, right? That's where you have tons of biofilm, and you have different redox conditions, and so you can really get transformations happening. It's basically, some people call it the river's liver. But it's basically like a big reactor for the stream. We also want to think in general about chemical fluxes, concentrations, where the reactions are occurring. So let's step through some of these things. And let me just give an example. Our group thinks a lot about nitrate removal, because nitrate is one of the most common things that impairs streams from a water quality perspective in the US and many parts of the world. And it turns out that the sediments and riverbeds are really productive places for nitrate removal if you can get the nitrate down there efficiently. And so what does that take? Where do we expect more nitrate removal due to denitrification, which, by the way, denitrification happens, for those of you who don't think about nitrogen cycling too often, it happens where you have less oxygen and more dissolved organic carbon. And so for that reason, if you can get the nitrate down into bed sediments where there's less oxygen, and if your sediment, the organic matter in your sediment, is a source of dissolved organic carbon, then you've got a nice little reactor there. So what do we want to have really efficient nitrate removal? We want more of the nitrate from the stream to follow the flow paths with the longer residence times. And what does long mean? It means long relative to the reaction time scale. So that should harken back to some of the things that we've been talking about with Dom Kohler numbers. Now the challenge though is that in nature the fastest flow paths have the shortest residence times. And another challenge is just that not all flows across the bed are hyporheic, and so these upwelling flows, for example, can be new inputs of nitrate. And we also want to know about nitrate transformations along those flow paths before that groundwater emerges within the stream. But we can sort of conveniently start by thinking about the hyporheic flow paths and what happens to stream water being essentially cleaned that's already in the stream. So let's step through and think about some of the things that we would want to know to understand this problem. First of all, we want to understand the fluid fluxes, so how much groundwater with a different chemistry is the stream gaining or losing. So that part that is not hyporheic. And the reason we want to know that is because when we have groundwater with a very different chemistry, because it's older, it's experienced different interactions, as it's come from the land with all of the human influence. We want to know how that groundwater with different temperature chemistry is going to impact the stream. And actually, I snapped this picture of Peter because this is a really nice example of thinking about this groundwater influence on the stream thermal regime in this case, which affects things like trout habitat. But you can also think about these processes affecting the nitrate concentrations that end up in the stream. We want to know about these hyporheic fluxes. And for this, I'm going to bring you back to some of this upscaled work by Jesus Gomez-Velez. And you can see with that basin scale model that they did, they were able to estimate the hyporheic exchange flux that you might predict along a river network as you go from high stream or low stream order to high stream order. And it's a little bit of what you might expect, but it's really fun to be able to show that with a model. Right? So as we go from high stream order with a rocky, more permeable stream bed and a lot of interaction of those shallow flows with a rough stream bed, a lot of bed form current interactions, we're going to have high fluxes. Notice the log scale here. And as we go to bigger rivers, we would expect lower exchange in part because of the changes in sediment texture and permeability of the stream bed. And I'm focusing here on the effect of bed forms, but there's also, of course, other elements contributing to these exchange processes, including flow through meanders. And if you're interested in learning more about that, Daniel Gonzalez-Duque has a nice poster here talking about the reactive transport through meanders. So this is how much exchange is happening, but we really only care about this exchange relative to how much water is moving downstream, because you can have really fast exchange through the hyporheic zone, but if your downstream transport in the river is like a fire hose, it's not going to matter. And so we really want to think about more than just the exchange fluxes. We want to think about how well connected it is. And a typical metric that we've used for that in the past is, throughout this community that I work in, is the turnover length. So the turnover length is basically an easy calculation you can do if you know the fluxes that says, on average, how far does river water go downstream before it enters the riverbed? And if you think about this turnover length concept going from small streams to big streams, you can see that actually it changes a lot within the basin. So small streams are incredibly efficient reactors that are turning over water through the hyporheic zone in a relatively fast way as it moves downstream. And in the lower parts of the basin, that river loses some of its efficiency, we think, following this model. What about the resonance time, though? Because that's important for thinking about reactions along flow paths. Again, they tend to have, the resonance time distributions tend to have power law tails, so it's almost comical to represent these power law distributions with one number. But we try, because at the end of the day, we want to say something about how long water sticks around in the stream bed. And so if you look at different studies, some people will use the median, some people will also think about variance or skewness, especially if they're dealing with field tracer breakthrough curves. to try to understand how long water is sticking around in these storage zones. But no matter what the number, if we try to look at the way this behaves in the landscape, you can see that as we go to bigger and bigger rivers, the water that does enter the stream bed, maybe it doesn't enter as efficiently, but once it's down there, it sticks around longer. And part of this is, again, related to sediment texture, the permeability of the bed. So now we can put all that together to think about really how efficient our stream bed reactor is, and talk about something that we call reaction significance. So the hyporheic zone will only affect stream water quality if the stream cycles through the hyporheic zone many times along the reach, and or some combination of the residence time also being long relative to the reaction time scale. So this is essentially a measure of cumulative exposure as you go from A to B. How many times are you jumping through the hyporheic zone and how long do you spend there? And so you can get transformations if you jump through the hyporheic zone many, many, many times, even if you don't stick around as long. Or you can make fewer jumps but stick around there longer and achieve a greater degree of chemical transformation. Either way. And so this is the reaction significance factor. Does anyone see a number that's been a theme of the past week in this equation? I haven't introduced these terms, so this is like a reaction rate. This is a residence time. And then this is your turnover length. Right? So it's the Dom-Kohler number, basically. It's the Dom-Kohler number. And essentially, it's a form of the Dom-Kohler number, right? It's how long do you have to react relative to the reaction efficiency, and then how many times are you skipping through that hyporheic zone. And we've done some studies where we've been really interested in understanding log jams in streams and their effect on the stream groundwater connectivity and the ability of the stream to retain solutes, retain nutrients. And what we have found actually is that the reaction significance factor, so how effectively the stream can transform any solute, it's going to be a lot greater in streams with jams than streams without jams, because these log jams that are formed by trees falling in streams or beaver activity, they create a really efficient way of pumping water through the stream bed because of those hydraulic head gradients. And so the reaction significance factor will increase at a given discharge, stream discharge, if you have more jams. And then as you increase the flow rate, the reaction significance factor decreases, and eventually the jams start to get submerged to the point that they don't contribute as much to the flow through the stream bed anymore, and it doesn't matter as much. Once you really submerge those jams, whether you have a lot of jams or not, the reaction significance factor is going to go down. But at low flows, base flows, these jams are essential for increasing the ability of solutes to be retained in the stream bed and available for uptake, ecological processes, things like that. So where in, for example, the Mississippi River Basin should more nitrate removal occur? It should basically occur, you would think, in the higher or the smaller streams, and it does. As we go to bigger and bigger rivers, right, we're getting essentially less transformation happening if we look at our reaction significance factor. So this just shows the power of small streams for transforming the chemistry of surface water. And you also see this sort of threshold behavior, which is very interesting. All of this, though, is from a modeling framework. So, obviously there's a lot of work to be done to understand how this relates to field observations, too. So let's take you now to the field for an example of why the chemical fluxes matter. So the chemical fluxes are something that we think about in terms of not just how much solute is moving across the bed, but we also want to know how heterogeneous that distribution is because it affects habitat complexity. So this is an example from some work that we did in a mountain stream in Colorado where we made a bunch of measurements of stream bed pore water chemistry over basically a two-dimensional slice through the stream bed. And we expect flow to be sort of from this up valley limb of the meander through the floodplain and to come out through this down valley end of the meander. And so therefore it makes sense that some of the higher dissolved oxygen concentrations that come from the river are found in this part of the stream bed where we get flow starting to enter through the floodplain. And then as we follow those hyperetic flow paths out, some of the oxygen should be consumed and we should see lower dissolved oxygen on this side of the meander. And then because this repeats higher dissolved oxygen, again on this side of the meander before that flow starts to enter the next bend in the floodplain. And we see that, but we also see a lot of complexity. And some of this complexity is due to things that you can see in this aerial image, like flow through pools and riffles. And so these are the types of features that drive oxygen in the stream bed that's available for things like salmon embryos, and at the subreach scale can shape where salmon would have their redds or their little egg nests. Another thing that we want to know is when you have a transformation, a chemical transformation happening near a sediment-water interface, where is the activity? Where is the transformation happening? So where are the hot spots? And when are they occurring, the hot moments? And so this is just a little example from something that we did in a river with fluctuating water levels. In this case, we were studying coastal rivers with tidal influence. And as that river level rises and falls, it pumps water in and out of the surrounding stream banks. And you can see here the changes in both nitrification, where there's oxygen near the stream and the water table, and denitrification, where a lot of these reactions are happening in heterogeneous sediments. You can also get a sense for when these reactions are dominating. And what you see here, the gray dashed line, is the river stage, the water level, going up and down with tides. In this tidal river, it's kind of cool because basically the water level looks flat most of the time, but then that pressure backs up from the tide coming in and you get a little bump in the river level. And then it stays flat again when the tide goes out for a while, and then you get a bump again when the tide goes in. Well, what that does is drives a little more nitrification when river water goes into the banks with its oxygen and its nitrate.
Andrea, what is the time scale of this movie?
So this is twice a day.
Okay.
Yeah. Sorry, there's no time axis here. So this was a semi-diurnal tide. And so when you bring that oxygenated river water in with the rising tide, you get a little bump in the nitrification rate, and your denitrification rate drops because some of that oxygen coming in will shut down your denitrification. And then as the tide falls, you're seeing some of the water that's been sitting in the surrounding sediments the longest, that's been interacting with sediment sources of organic carbon. And you start to get this, and the oxygen's been consumed, and you start to get this rise in denitrification again that increases up until the next tidal wave comes in and brings in new oxygen. So these are just some of the things that we think about to really understand the chemical transformations happening near sediment-water interfaces. Let me show you a little bit more of an example where we've done quite a lot of work, not just on the chemistry, but also a little bit of microbiology with the help of Mike Wilkins at Colorado State University. This is from the East River site in Colorado, and we wanted to understand here how hyporheic flow drives pore water chemistry and microbial community composition, and how these processes could change because the hydrograph is changing in a changing climate. Spring snowmelt is changing. That cycle of spring floods is changing. So we wanted to understand these processes over space and time. And so this is the site I mentioned before where we worked on one representative meander. We hope it's representative. From the East River. And we made a bunch of measurements of streambed chemistry and microbial community assemblage at the same depth of about 20 centimeters. As I mentioned before, we sort of expected this flow because of the way the river interacts with the meander going around the bed. We expected this longitudinal flow to affect the chemistry in this part of the reach versus this part of the reach. And the sampling was a lot of fun. This is just giving you an idea of the field site. We collected samples into these really cool filters that could be used then for microbial analysis. And we also made some measurements of the vertical head gradient in the stream bed. This is a very technical measurement where you pound a pipe into the bed and you look at the water difference inside of the pipe and in the stream surrounding it. And so the pipe is open at the bottom, so the height the water level rises to in the pipe is telling you hydraulic head at this point at the bottom of the pipe. And then the river level is telling you hydraulic head in the surface water. And so based on whether the water level in the pipe is higher or lower, you can tell whether there's potential for upward or downward flow in your stream bed. But it doesn't give you velocities. So one other cool thing that you can do is some temperature sensing to use heat as a tracer to figure out the upward or downward flow, basically because convection, thermal convection, is going to affect the way temperature signals propagate upward or downward in the riverbed, and you can use that to figure out the flow. So, let's see here. So here are some of the things that we found in terms of just patterns of vertical flow and mixing. This is showing you the vertical hydraulic head gradient in the bed. So the greener and bluer colors are downwelling flows, and then the yellow and orange and red tend to be more upwelling zones. And then with that, you can also look at the percent surface water. For this, we just did an n-member mixing calculation looking at specific conductivity because groundwater at the site has a slightly higher specific conductivity than stream water. And so we see these patterns of, in general, changes in upwelling and downwelling as you go through riffles, and then some changes from side to side as you go around the bend that affect the amount of groundwater that tends to come out. And so we get these patches of groundwater coming out on this end as we go around the bend. And this is what affects the dissolved oxygen I've already showed. We can also do like a really simple trick where we look at the dissolved oxygen in the stream, and we look at the dissolved oxygen in all of our pore water samples. And some of the samples that were most representative of our groundwater end member have pretty low dissolved oxygen. The lowest we can measure when we pull a sample out and try to quickly measure dissolved oxygen. It's probably much lower than this because we expose the sample to atmosphere the minute we pull it out, but it gives us an idea. And you can see how many of the pore water samples don't follow this conservative mixing line because there's oxygen being consumed in the stream bed due to, for example, aerobic respiration. We also looked at the dissolved manganese, and this one followed maybe a little bit less of an obvious trend, but some of the more groundwater dominated samples have really high dissolved manganese concentrations because it's reducing groundwater. The stream looks like this, and then the pore water is kind of all over the place, but a lot of the pore water is actually depleted in dissolved manganese. And if you walk around some of these sites, you can actually see manganese oxides forming in the stream bed. So a lot of this can be due to dissolved manganese from reducing groundwater mixing with oxygenated surface water as it moves through the hyporheic zone and precipitating out manganese oxides. And then we also looked at dissolved organic, we looked at a lot of things, but I'm showing you the like greatest hits. We looked at dissolved organic carbon. We also looked at its quality, not just its concentration using fluorescence, but I'm only gonna show you concentration today. And you can see again, it's a little bit all over the place in terms of the pore water, but to me, this is just an indication of how heterogeneous these environments are. And I've never worked in a place before where the sediments weren't a really important source of dissolved organic carbon. So some of the areas with higher dissolved organic carbon, I would bet it's coming from the sediments. And then we also looked at the streambed microbiome. And this was interesting. So this is Shannon's H versus percent surface water. What is Shannon's H? It's basically a measure of diversity. And for those of you who have thought about entropy before instead of ecology, it's like a measure of entropy of your stream bed, of your microbial assemblage, right? So the more species you have that is incredibly diverse relative to the stream. We looked at how that assemblage changes. Different samples are actually diverse in different ways. I won't show you that data today, but they're not all the same. Some parts of the stream that have more of a groundwater influence have a different assemblage, but in the end, still similarly diverse to parts of the stream that have more of a surface water, stream bed that have more of a surface water influence. And then another example from some work that I was a collaborator on at a totally different site, we looked at the depth profiles in the microbial diversity. And what you see here, so this is a sort of messy plot, but I would tune your eye here to this blue line, which is percent river water, ranging from 100% to 0% as you go down with depth for these pore water samples. So here's your mixing zone, and here's where it's all groundwater end member down here. And if we look now at Shannon's H, so microbial diversity, you can see that the highest microbial diversity is actually in this mixing zone, which is super cool. And so the stream is less diverse, the groundwater a little more diverse, but like not a lot so, and then this mixing zone is where the community really has a lot of variability. Yeah?
Question about that. Do you think you would get different answers at different flow conditions?
We do. Yeah, and so Bob Dangsack has done this work actually in 3 different seasons. And you can see these differences for 3 different seasons where we have slightly different profiles in the pore water chemistry. But the overall trend is still very close. So I would suggest that even though this mixing zone is very dynamic, the peak in diversity is still kind of persistent over the year. Who knows over longer time scales, like erosion. I mean, I think this is a really interesting area for research, thinking about these changes over time, and not just who's there, because right now, and in fact, in this talk, I'm only showing you, any microbial stuff I show you is really just looking at who's there and not what they're doing. But that's an exploding area of research as well. And so we also wanted to understand how this affects reactive transport processes that we care about, these different changes in mixing within our field site. And one of the things that we noticed were big changes in the dissolved manganese concentrations over time at our site. We care about manganese oxide formation and the manganese cycle, because manganese oxides are like sponges that can be sites of absorption for other metals that affect things like health. And then in extreme concentrations, manganese can be toxic. The part of the stream that we were working in is more pristine, but there are a lot of mining activities at different areas within this basin in Colorado, and there the manganese chemistry can be very different and important in different ways. So we wanted to understand the behavior of manganese in the stream bed, at least at this site. And our idea here was that when we have stronger upwelling during the summer base flow season, but before you get too long into fall, we should get this reducing groundwater with a lot of dissolved manganese upwelling, interacting with river water with its oxygen in the shallow stream bed and forming lots of manganese oxides. And then when we have the spring snowmelt, that's when there's a lot of course, like exchange. Things are happening really quickly. And we can actually get some downwelling occurring. And we wanted to know, as oxygen comes in, but also dissolved organic carbon from the river water, are we going to get more manganese oxidation or reduction? What's going to happen to those manganese oxides? And so for that, we did some sampling campaigns over an annual hydrograph, and sampling on this day was a little scary. It was a raging river, so my apologies, we didn't get the real peak, but if we had, I don't know if we would have survived. It's a beautiful site. Again, we use temperature sensing to understand changes over the whole year when we weren't out there sampling in the vertical fluxes. And the idea here, I'll unpack this just a little bit more. The idea here is that you can use smaller timescale fluctuations in river temperature that penetrate into the bed to say something about whether there's a strong signal of convection carrying that signal down faster than you would expect by conduction, or whether there's upwelling sort of suppressing that penetration of the temperature signal into the bed. So if you were to take temperature measurements at different depths in the stream bed, you would expect in a downwelling zone all the depths look very close to the temperature signal in stream water. If you're in an upwelling zone, you get more of that stable temperature signal coming up from below. John mentioned the other day that stream temperature signals are not sinusoidal, and I fully agree. But for the purposes of just demonstrating the concept, I show them here. And then this is our data over the year. And I'm only just going to show you one site here, the B site, which was more representative of an area with a little more groundwater influence. So what are we looking at? This orange line is the river discharge. So here's our spring snowmelt peak. And then this is our Darcy velocity upward or downward within the bed. And so as we go through the summer base flow season towards the winter time, we get a fair amount of base flow and vertical discharge, like vertical upwelling of groundwater in our stream bed. And then around this time of year, things freeze up. And we just kind of assume that nothing's happening. All the temperatures are zero, so we can't really tell anyways. But it's frozen. And then it starts to thaw like all at once, and here comes the spring snowmelt. We see the thawing in the riverbed and our temperature signals, and we get a little bit more groundwater influence coming up during that time. But as we get this big snowmelt peak, we actually lose our data because our thermistors are getting ripped around. But then we get them back in, and we see that there is this time where there's this reversal of flow, and we actually get downwelling into the sediment associated with basically this change in river stage and all of the things that are happening in the floodplain as well. And so how do we model manganese transport in this system? Well, it turns out that a 1D model is often like pretty sufficient in a lot of the field sites where I've worked, because we're limited in data in the field. And so we went with a 1D model. But in this 1D model, we know the flow upward or downward in the stream bed. We measured it, or we figured it out with temperature sensing. What we don't necessarily know is the effect of basically mechanical dispersion, but also giving a nod to Peter Kong's work. Like there's a lot of effects of turbulent momentum transfer in these streambed sediments. Inertial effects are really important here. So we need to figure out what our effective D should be for this problem. Fortunately, there's actually some really nice experiments that tell us a little bit about that. And then we can try to do our best to constrain RR2 from our data and from the literature. But once we have these things, we know the boundary conditions at the stream bed, they're dynamic, we can plug that into a 1D advection, dispersion, reaction equation, and we know things like specific conductivity, we know like the dissolved organic carbon in the stream, that becomes our boundary condition for this dynamic problem. So how do we get at that D? It's interesting. There's been a lot of, I wouldn't say a lot of work, but a little bit of work on this from a flume perspective. And it turns out there's 3 regimes as you go from a low permeability Reynolds number to a high permeability Reynolds number, which is defined here. I'll talk about that in a second. There's 3 regimes. There's a regime where the effective dispersion coefficient is constant. It's just driven by molecular diffusion. And then there's a regime where basically inertial effects start to dominate. And then another regime where really turbulence is important here. And so what is the permeability Reynolds number? It depends on the permeability of the stream bed and the shear velocity in the surface water. And so this is really giving you a sense of how things are mixing with depth in the stream bed as you get more and more vigorous sort of current bed interactions for different permeabilities. And then the Schmidt number is also important here. The Schmidt number is just another really nice dimensionless number that is balance between the diffusion of momentum versus diffusion of mass. So it's telling you about the fluid fluxes versus the solute mass fluxes. And so we can actually use these scaling laws that have been worked out for flumes to predict D over the year. We know shear velocity in the stream just based on channel hydraulics. And so we can put together these models that match our data pretty well. The only thing that didn't work so well was oxygen, but that's because when we pull the sample out, it has higher oxygen because we're exposing it to air. But otherwise, things looked pretty good. And what we can do then is try to understand how mixing in the stream bed occurs vertically and over time over the year. And you can see when groundwater is upwelling, we get these. So specific conductivity, this is more river water-like, this is more groundwater-like. You see the mixing profile over most of the year when groundwater is upwelling looks like this. And then when the stream bed thaws after this frozen period here and the river discharge increases, we get this huge flux of solute into the bed. And then a return to more base flow conditions at the end of the season. And we can use that to say something about the manganese flux between the stream and the sediments. And so this is just showing you pretty constant when we're in that base flow regime, but then we get this huge flux of manganese to the stream during the hydrograph peak. And we can also look at the balance between precipitation versus dissolution of manganese oxides and see that actually the hydrograph peak, I would have predicted maybe more oxygen, more manganese oxide formation, but it turns out that the dissolved organic carbon is a really big part of the story too, and that you get reduction of manganese oxides during this period where the stream flow is ripping downstream, and then a return to the formation of manganese oxides. Okay, how are we doing? I'm gonna now take you from a stream site to a coastal site, and I wanted to just at least spend like 15 minutes talking about coastal processes because we're here and it's beautiful. So hopefully I inspire you a little bit to think about these exchange processes when you go out and are sitting on the beach doing, I don't know, beach geophysics or just fun leisure time on this trip. So this is sort of a conceptual model of all the different ways that surface water and groundwater mix in coastal aquifers. And it's a little bit more complicated than this, but basically you have a freshwater lens that sits under land, and then salt water that mixes or that comes in and sits below the surface water, and they mix in an interesting way that drives all kinds of cool reactions. So one of the things that we want to know when we're thinking about chemical fluxes is how much of the water is coming from land to sea, because the chemistry of fresh groundwater coming from the land, where humans live and where the ecology is obviously very different, is going to be different and affect surface water quality along the coast if we have enough of that outward flow in a concentrated way. But there's also all kinds of, so I just talked to you about the hyporheic zone and surface water recirculation through stream beds. There's all kinds of recirculation of saline water through the seabed as well, and over a variety of different length scales and time scales. So right at the surf zone, we're going to get circulation of salty water right where the waves come in and out and lap up. And then out in the seabed, Charlie Harvey mentioned the importance of critters doing hydrologic things. There's all kinds of critters that live in the seabed that pump water in and out, but there's also non-biological things happening. So basically deeper flow paths that come back in, and then salt disperses across this interface, and this process drives circulation through the stream bed, or through the coastal aquifer here. So a lot of different processes that drive mixing. What's the balance of fresh discharge versus salty discharge through the bed? You can imagine that matters a lot for chemistry, things like nutrient recycling, nutrient regeneration, and then also contaminants moving from onshore to offshore. It turns out that the fresh component of discharge offshore we think is about 1% of the total fresh water flowing from rivers to the coast. So for every 100 units of river discharge, we think that there's about 1 unit of fresh groundwater discharge along coastal zones, and then that the saline fluxes are hundreds of times more than that. And all of this is really important for different ecosystem services. How do we know these numbers though? So our group has worked a little bit on trying to get at the fresh flux over continental scales, and we used, if I might say so myself, a stupidly simple method where we basically just looked at all of the little catchments in the landscape that don't drain to a river. And right now I'm talking about surface water flows, but under some pretty strong assumptions, you can assume the groundwater flow paths are similar. And so we found these little catchments. We looked at estimates of what the recharge might be in those catchments based on global recharge models, which there's a nice poster about global recharge models too out in the courtyard. But that gave us what we think the freshwater discharge should be right at the coast. And if we do that over global scales, we get a distribution that looks a little bit like this, where fresh rates of submarine groundwater discharge are highly variable around the world. No surprise, the dry regions have less discharge, the wet regions have more. One thing that's interesting that I'm not showing you is we actually also see differences across tectonic settings because of the way that catchments, the topology of catchments in steep terrains versus more gradual terrains, and the way that funnels fresh groundwater to the coast. But over 40% of fresh submarine groundwater discharge occurs in the tropics, and that is not necessarily where a lot of the studies come from, so it also helps us think about where we need to do more field work. So this plot here shows you the distribution with latitude of fresh submarine groundwater discharge, and a lot of it is occurring in wet areas near the equator. How else do we try to estimate these fluxes? There's all kinds of ways to get at it in the field, including natural radiogenic tracers, seepage meters, different direct measurements with wells, and then geophysics, which we've got a little bit of a taste for in this, these 2 weeks. Just a quick overview of the natural radiogenic tracers. There's many, but these are a common tool for understanding water moving through sediments. Why? The idea is that these are coming from decay processes that happen within the sediment. And so if you measure them in the surface water, you're seeing basically the products of what has happened down in the sediments before that water comes back out again. It's complicated though, like all things, because sources of these radiogenic isotopes include groundwater that is discharged to surface water and is now entering your bay or your coastal zone. Of course, groundwater, that's what we care about trying to measure. But there's also diffusion happening right near the sediment-water interface. Should we count that as submarine groundwater discharge? And to what extent is that contributing to the inventory of radiogenic tracers in our surface water that we might measure at any time? There's all kinds of also exchange between other surface waters, like through inlets in a bay, or just with open sea or open ocean. So when people do budgets to try to understand how submarine groundwater discharge is contributing to the radiogenic isotopes that they measure here, it requires a little bit of assessment of what these different inputs might be. So one of the things that I like to do, therefore, is combine this work with direct measurements with seepage meters. These are really simple. It's a drum cut off at the bottom. You stick it down in the bed, and you put a bag on, and you measure how much groundwater comes out over a certain amount of time to fill the bag. And it's really simple, but at the end of the day, it tells you what's flowing up or down through the bed, because you can pre-fill the bag a little bit too and look at the water going out and leaving the bag if there's recharge. And it's subject to all kinds of other uncertainties associated with whether the bag is being tossed and turned by the currents and things like that, but it's one other tool we can use. And you can also measure whether the water that enters the bag is fresh or salty, and think about how much of that water is fresh or saline recirculated water. There's also a lot of geophysical techniques that are really nice for getting an idea of where the fresh groundwater outflows might be, because fresh groundwater has such a big effect on bulk resistivity. This is just an example of one approach with marine continuous resistivity profiling, where you're basically towing everything behind a boat instead of what some of the teams have been doing on the beach. Here's what can be done on the beach, and there's a team who's doing some of this work right now. It's really exciting to see the results that you all come up with. These are not those results, so these are from a different site. So let me show you how these methods are now being combined as part of a broader project that's funded by the Spanish government called Mucho Gusto, trying to understand submarine groundwater discharge at different sites with different geology, including a site that has some karst conduits, as well as a more sort of traditional alluvial geology kind of site. And so this is the site with more karst type of geology with focused springs, and there's a bunch of different transects where the team at UPC that I've been collaborating with has made some really cool measurements of seepage rates, electrical resistivity surveys, and other measurements. Here is some of the work on both the marine and the land-based geophysics. And what you see here is a slice of the estimated resistivity over this area offshore. And so that's all at the same depth, and that means that these areas with higher resistivity here are associated with fresh groundwater being near the seabed. If we look at cross sections going along here of the resistivity inversions, you see these really cool zones of fresh groundwater discharge and then focused upwelling offshore that are presumably related to karst conduits or greater connectivity in the surface due to heterogeneity. And these circles here are also showing you seepage rates. So the areas with the bigger circles have a greater total submarine groundwater discharge rate, but where they're filled with bigger black dots, that's because of more fresh water contributing to that seepage. And so it's no surprise then that there are some places that are more resistive where you would expect fresh groundwater, and there's a big component of the submarine groundwater discharge that we measure as fresh with our seepage meters. You see other areas like this where it's more salty, as we infer from the geophysics and with the seepage meters. And it's not perfect one-to-one, but there's some pretty good correspondence here. We can combine that with sort of a conceptual model and some geochemistry here, and I won't spend very long on this, but the idea here is that the zones of focused groundwater discharge that we see in geophysical inversions have a very different chemistry than zones of more sort of diffuse, salty water recirculating through the sediment versus the submarine springs here. And so all of these processes influence the nutrient flux to surface water that is important for thinking about surface water quality and marine productivity. The other thing I just wanted to show you, this is from a different site that is part of the Mucho Gusto project, but as we follow these flow paths from fresh groundwater offshore, we encounter salt water, and there's mixing between fresh water and salt water, and nitrate concentrations along those flow paths tend to drop in a way that's not conservative. So this interface between fresh water and salt water is really important as a zone of chemical transformation. And for that reason, the marine folks have called these mixing zones the subterranean estuary, which is maybe a sort of a different way of thinking about it for physical hydrogeologists like myself. But if you're ever in an estuary on the land surface where a fresh river is coming out and salt water from, say, the ocean is mixing, it's an incredibly ecologically productive place, right? With it's habitat for different fish. It's a place where a lot of transformations occur in the water, and you get all kinds of crazy processes happening with flocculation in the water column. Well, the idea is that in the subsurface where we have these mixing zones, we also have a lot of transformations occurring, and it can look different in different types of geologic settings, but arguably most of the work that's been done has happened in these sandy coastal systems, and there's much more work to be done on heterogeneous environments. I was part of a larger metadata analysis where we looked at chemical transformations along mixing zones between freshwater and saltwater. And the idea here is that if you plot something like a nutrient versus salinity, and there's no reactions along flow paths as you go from freshwater to saltwater through the mixing zone, you should see a line that looks like this. But if you're getting something like nutrient regeneration, production of, say, ammonium, that line is going to bend upward. Or if you're consuming something, say, like nitrate due to denitrification, that line is going to bend downward. And so the team that I was a part of grabbed data sets from researchers all over the world to look at how reactive these mixing zones are. And what they found is that a relatively small portion of all of the sites that they looked at were conservative with respect to nutrients in different form. But a lot of them were productive or consumptive, and so there's a lot of work to be done to figure out why the subterranean estuary might be, say, a producer of nitrate in one place versus a consumer of nitrate in another place. But this was one of the first times anyone had tried to look at how transformative these environments are in a holistic global sense. Okay, I know, let's see, we have just a little bit more time talking about mixing zones. This is one of the biggest mixing zones that form in a coastal site between the freshwater lens and the saltwater that's recirculating underneath the seabed here. It never looks this perfect. It usually is highly interfingering due to geologic heterogeneity. But this is an important area in the subterranean estuary. And we can actually try to estimate the depth to this mixing zone. If we have time, I'll do that in just a minute. But there's other mixing zones that happen at smaller scales due to all kinds of different processes that I love to think about. So bed form current interactions, wave pumping. Even if you have a flat bed, the interaction of waves with that flat bed drives exchange across the bed, and then things like critters. There's all kinds of different processes that drive this. There are some nice mathematical models for calculating the fluxes due to these different processes. So one example I'll show you here, just really as an example, is the model for wave pumping, where exchange increases, of course, with hydraulic conductivity, that thing that varies by orders of magnitude that we can't escape as hydrogeologists. It increases with wave height, so bigger waves. It does something non-linear with wavelength, right? The idea is if you have really long waves, that's taking away from your hydraulic head gradient driving flow. And then if you have really, really short waves at sort of the end member limit, then you're not going to get as much, like basically you're not going to be able to get things through the bed as easily. And then in the middle is that sweet spot. And of course only certain combinations of wave height and wavelength can form at a given water depth as well. But as the water depth goes down, those waves are going to interact with the bed more. And so when you have shallower water depths, you're going to have more exchange. And we can use this again to upscale and think about exchange rates across. So this is offshore of the southeastern US. Looking at water depths and typical wave conditions and permeabilities to try to understand fluxes across the seabed. Why does it matter? Our group has worked on this problem for thinking about algal toxins, because in Lake Erie we have waves, we have permeable sediments, and we have terrible harmful algal blooms. And so when the algae blooms, sometimes they produce toxins, sometimes they don't. But when they do produce toxins, we wanted to know, does exchange through permeable sediments help with degradation of the toxin and lead to toxin removal? The lake bed essentially acting like a filter. So we ran some experiments in the lab in a tank with an aquarium wave maker. And we used some blue dye to just map exchange through the sediments under our wave conditions. And it's really fast, so this was only 3 minutes in. You can see exchange down to like 5 centimeters depth, not just molecular diffusion. And we looked at what happens to an algal toxin when we add it to the lake. And so what happens when we introduce the algal toxin with a conservative tracer chloride? Initially the chloride drops and then levels off once a sort of equilibrium mixing is achieved. But the microcystin, which is our algal toxin, keeps dropping due to degradation in the sediment. And the pore water profile will look something like this if degradation is happening. So did we see that? We did. So what we saw is that concentration dropped with the chloride and then leveled off, but the microcystin, this data is a little bit noisy because of the way we were analyzing it, but the microcystin concentration dropped substantially over time due to that breakdown or removal in the sediments. And we can look at the pore water, and it does what we would expect. So here's the chloride concentration in the pore water. It's fully mixed with surface water when the two are the same. In the algal toxin concentration, though, it first increases as that mixing occurs, and then it decreases due to degradation in the sediments. And so we could again put together a mathematical model with a little enhanced dispersion coefficient. And what we see is that as we go to more permeable sediments or to basically bigger waves, we get a lot more removal, and we quantified that removal. Basically, the World Health Organization recreational guideline is that you don't want to swim in this water if there's a microcystin concentration of more than 20 parts per billion, which happens, we surpass those a lot in Lake Erie. But it turns out that a sandy lake bed can remove half of the equivalent water column mass just in tens of hours. And otherwise, you would have a really persistent amount of toxin in the water. So we think this situation is important in coastal areas where people want to recreate. Just a little ecosystem service. Okay, so now with maybe like a remaining 5 minutes, I just want to take you to our field site right out here. I'm sorry I can't take like 90 people out to the coast, but I want to just think a little bit. What's happening on this beach? Where are there mixing processes and reactions? So I first want to ask you all, if you were to go out to this beach and do some geophysics, or stand here, how would you figure out depth to groundwater and how much fresh water there is, right? Because that's an important thing to think about for understanding where mixing is occurring between freshwater and salty water. Also, if you're a local who needs a groundwater well, you don't want to put your well in salty water. So for you guys, if you were to step out there, how would you estimate these things? A well? What if we could like do a thought experiment and we don't have a drill rig, but we want to just do this without a well? Who thinks that if I go 2 meters down, it's going to be fresh? Like right here. Raise your hand if you think it's fresh versus salty. Fresh? Who thinks salty? Okay, we have a mix. So let's do a quick calculation. So I would assume that if I dug a hole on the beach, and you guys could dig a hole on the beach, you probably need to go, what, like, here's the water, here we are, maybe like a meter up, maybe like a meter to hit fresh water, right? Right? And then we want to know how much deeper is it going to take to go until we hit salt water. So let's do a little calculation, right? So let's say we're sitting here, our fresh water is here, we've got like a, basically, I don't know, let's say that we go like half a meter down and we get to the water table, and then let's say we go another meter down and we're basically at sea level. So let's say the water table is about a meter above sea level at this well. How deep do you think this interface is, right? It's actually something we can calculate if we make some stark assumptions, including, first of all, the assumption that everything is balanced, no flow, and second of all, an assumption that we have a sharp interface here. But we can calculate this if we just assume that the weight of water here balances the weight of water out here, or basically that the hydraulic heads are the same, equivalent hydraulic head, right? So we can say, let's call this H equals 1 meter, so that's the height of fresh water above sea level. And we want to know D, right? So how much deeper below that is the saltwater interface? So let's call this D. If we want to just balance these two columns of water, then here, let's make it easy. Let's compare 2 points here and here with like a hydrostatic condition, right? And so here we need that the weight of the fresh water, density of fresh water times gravity times this hydraulic head above sea level versus the fresh water below, this whole column thickness. This has to balance over here the salty water, right? And then let's see here. So here we've got D over here because sea level is flat. We can rearrange this equation to figure out how H and D are related. And so if we do that, right, so let's see here. We are going to get right, and then let's see here. Oh, did I do this? I might have done this wrong. What's that?
Gather your D's onto one side, probably, and then divide.
Yeah. Thank you. Rho F G H. Right. Rho S G D minus rho F, G, D, right? And then we can of course cancel out our G's. And then if we do all that, we can relate our H. It's just going to be equal, well, we can actually, maybe it's easier if we do it the other way, rho S minus rho F over rho F times D. And if we put in typical values for the density of salt water and the density of fresh water, you can do this, right? I'm kind of zipping through it now because we're running out of time. But it turns out that D is just on the average 40 times H. So what does this mean? It means that if we're sitting here and we've got maybe like a meter of fresh water head above sea level, we would have to go another 40 meters down to get to salty water. That's kind of crazy, right? It's like more fresh water than you would think. But on the other hand, nothing is this simple. You've got dynamic mixing. You've got all of these processes, right? And then on top of it, if you were to start pumping a well here, basically if you draw down the head by a meter, you're going to draw up that saltwater interface by 40 meters. So it's really easy to get the breakthrough of salty water to your well. Let's think about some other things that are going on here. So I walked around and here's some fresh groundwater coming out. What do you guys see here? Like, do you see any signals of what might be going on with the chemistry? Yeah. So what is the chemistry, or like what is probably the chemistry of this groundwater? It's reducing, right? Yeah. And probably has some iron, I'm guessing, right? When it hits salt water, there's some really interesting things that are going to happen there. If the salt water it has a lot of oxygen in it, if the salt water has dissolved organic carbon in it, right? And so these mixing zones are very important for this reason. Another thing that happens if you walk around this beach, right? I love these, just looking at the way the seepage face changes as the swash goes back and forth. And so what you see is sort of as the water level goes down with a wave, the seepage of salt water back out, right? So salt water comes in when the wave goes up, and then as the wave recedes, you see the seepage and salt water going back out. And so this constant swashing is recycling water through the beach face and allowing interactions with biofilms and transformations to occur. And we can model that. Oops, I forgot the reference. I think that's Claire Robinson's work. Oh, no, it's there. So you can model that and think about reactions that happen there. And then, of course, if you stand up on a rock and look just right between the waves, you see these bed forms forming, like these ripples, really beautiful. And you think about all the waves that are driving exchange through the bed and how that's affecting the transformation, so the nitrogen cycle, for example. And we can model that as well, which is super fun. Okay, so what are some of the things that I think are frontiers and opportunities? Charlie Harvey mentioned the catfish swimming through your porous media in Borneo, right? And he said, I've never seen a field site where it didn't seem like the biology, the organisms are important, even like from a physical hydrogeology perspective, and I would agree. Working in riverbeds, working in coastal zones, and I think that there's a lot of opportunity to really understand the effects of the organisms more on these exchange processes and these reactive transport processes. I also, being here and learning a little bit of open foam and seeing some of the cool research that is happening where inertial effects matter, I think that open framework gravels are something you find in all kinds of aquatic environments and require a lot more study. Like if you've ever been standing sort of in the beach or in a stream bed, you see how biofilm grows on these big cobbles. And these are important reaction sites that are affecting transformations in the bed. And due to excess nutrients and all kinds of other things that humans do to the environment, we really change the flow through these spaces as biofilms grow. And that has feedbacks for the way that these aquatic sediments act as reactors. And then just another thing that's low-hanging fruit, I think that there's a lot of work that still needs to happen in the coastal zone to reconcile sea-based measurements with radiogenic tracers, with land-based hydrogeologic methods, and to do that in places where the geology is complex, where there is karst, for example, and places that have unique chemistry as well. And so what this study here is showing is just that most of the work that we've done on coastal surface water groundwater interactions and fresh or in submarine groundwater discharge has happened in typical estuary, bay, lagoon type of settings with sandy sediments. So there's a lot more work to be done here. And with that, I just want to give a shameless advertisement as well for any of you who are interested in either continuing in the community of coastal surface water groundwater interactions or breaking into it. There's an exciting conference coming called the SWIM meeting that will take place in Barcelona in June of 2025. And I'm part of the organizing committee for that, but the lead organizers are my collaborators at UPC. And then also there's a cool session on surface water groundwater interactions that's a catch-all for river and coastal and marine environments at AGU this year. So feel free to check those out. And I can take any questions if we have time.
Can you go back to the slide where it was on the water in Tages, but like with the ripples?
With ripple, there's a lot of ripple slides.
No, but it was specifically the Tages, your observations in Tages. And the next one? This one?
Yeah.
How is that evidence of saline groundwater discharge?
Well,
Specifically, what observations are you using to say that?
I mean, it's a great question. Ideally, we would want to make some measurements, but the coast is like over here.
Yeah.
And it's saltier. Like basically, we're a little bit offshore, and actually, most of the time, fresh ground. If you don't have karst or fractured bedrock, the freshwater discharge is right at the basically low tide, low wave base line, and so I'm below that. And below that, you start to get into the sun. But I don't know. I mean, I would love it if maybe the geophysics folks want to go do some geophysics running offshore as well. I don't know. We could try to drink it. I don't know. Yeah.
I've got a question for you. When you were talking about rivers,
Yeah.
going around that large meander.
Yeah.
When water goes onto the floodplain, are you expecting exchange with the soil there?
Yeah.
The water that re-enters the river, has the chemistry substantially changed?
Yeah, so most of the hydrograph year that you were looking at, the water was still confined within the channel. But when water does overtop onto floodplains, I think there's got to be a ton of carbon exchange. And also I want to point out that like when water's flowing through meanders, it's not just one long stream tube that doesn't interact with anything else. So you've got more carbon shallow in the floodplain, most likely, just because of the way soil profiles often look, although floodplains can be messy. And then you've got a water table where there is really efficient oxygen exchange. And that's some other work I've done that I haven't showed here with folks at UPC and CSIC. But that water flowing through the floodplain is going to, if it's near the water table, potentially entrain more oxygen, but also more carbon if it's interacting with carbon-rich soils. And it doesn't have to overflow the floodplain for that process to happen. But when you do overflow the floodplain, I think that there's got to be some really effective penetration of more carbon-rich stuff down into your aquifer that's resetting the chemistry on these flood events. It's a great question. Yeah.
Thanks for your nice presentation. I think there's a slide where you show up the dissolved organic carbons in the surface water.
Oh, yeah, yeah.
Yeah, but how do you distinguish it from the inorganic carbons in the surface water?
Was it the map of dissolved organic carbon here?
Yeah.
Okay.
How do you do that? I mean, because in the surface water, there will also be the inorganic one, right?
Yeah, no, we are just taking filtered samples and then we run them on the Shimadzu TOC analyzer, and that's, I'm not an analytical chemist, but...
Do you think there's any way you can only measure the inorganic carbon in the sample?
Sorry.
Do you think there's any way to only measure the inorganic carbon in the sample?
Oh, yeah, but you have to take the sample in a different way too that involves like no headspace and things like that, if I remember right. We haven't sampled for DIC in a while, but it's a different sampling protocol and then different analysis. Yeah.
So I know that saltwater and freshwater can mix on the surface and it makes brackish water. Why is it not mixing underground? Why is there like...
It does. So actually, if you have fresh, and I think this gets to the first question about how do we know that it's salt water circulating through the sediments. I mean, I think circulation through the bed, when you're mixing surface water through the sediments, like it's salty water that's sitting up in the bed. I mean, salty water that's sitting up in the water column mixing through here, but the question is what's underneath. And if you're right at sort of what I described as like low wave base, you can have fresh water upwelling and being overlain by salty water. And we've actually done some models too looking at the buoyancy effects of that process. But it depends on where you are in this overall zone. Where is it? This overall zone, right? So if you're in the swash zone, any waves that come in are mixing salt water with more salt water, because that's just different scales of salt water mixing in this zone created by swash and tides. If you're here and there's some waves interacting with the bed, it's salty water over fresh water, and you're going to get buoyancy effects too, and we've done some models of that. And then if you're out here, then it's just salty water over salty water, but still different chemistry. It's like the hypereic zone, right? In the hypereic zone, it's fresh water over fresh water, but still with different residence times, different chemistry, different interactions with organic matter that still lead to really cool transformations that happen in these mixing zones. I hope that answered it, yeah.
Hey, Audrey,
Oh, over here. No, sorry, I should listen to my...
There was a slide where you showed the maps of the denitrification and nitrification.
Yeah.
I was wondering if this kind of modeling we could use for infiltration basins or manage aquifer recharge systems.
Yeah.
Can you control the infiltration rate?
Yeah, so that's a great question. And actually, I think one of the really fun things about collaborating with people at SESIC and UPC is there's a lot of interest in managed aquifer recharge, and they're interested in a lot of the same processes of what happens when you take something with one chemistry and you move it down into an aquifer and potentially through these different organic rich zones as well, because you build up some nice organic stuff and manage aquifer recharge near the surface. Like we have the same questions here in our natural kind of recharge discharge scenarios, and all the same modeling techniques are the same. The geometry is just a little different. Yeah, it's a great question.
Hey Audrey, thank you for this amazing presentation. I have a question on the site that you have, where you have upwelling and then suddenly you start having downwelling and processes there.
Yeah.
If that is happening in the bed form,
Yeah.
and most of the year is actually upwelling, are we maybe overestimating the amount of
Yes.
processes in bed form?
Yeah, so that's a great question. And if you have really strong upwelling or really strong downwelling, it will swamp the effects of these kind of recirculating flow patterns. Just by superposition, you can think about it, right? If you have a flux of like, I don't know, a centimeter per day through a ripple. That's not a good number to throw out there, but then you have like an overall rate of, you would have a flux of a centimeter per day through a ripple, but then you have an overall tendency to have groundwater downwelling at 10 centimeters per day. You're not gonna form those exchange flows due to bed form current interactions. Those will get swamped. And I think that does happen in a lot of places. Like I did some work in my PhD where we wanted to understand the effects of logs in streams because of these temperature effects, and we added logs to the stream and we saw changes in the thermal signal in the bed because of the recirculation around the logs, but it was a much smaller footprint than we were expecting because we were in an overall losing reach. But that's still important to think about too, because even if your exchange is smaller, we were working in that stream because they wanted to support a fishery. And they were really interested in this question of how to get more like thermal regulation, kind of buffering in the stream. And so they were in this mountain meadow stream, lots of sun, trying to support brown trout, and all the groundwater is downwelling. So there was not good cold patches in the stream, but then you put a log in, and you create even a small exchange zone around that log that's smaller than it would have been because of the downwelling, and you get like a little zone where you create a different temperature condition. And so I think even when it's a little bit restricted due to the overall gradients, cool things can still happen unless you're in a situation where it's totally swamped.
So maybe one last question.
Thank you, Audrey, for the presentation. I think that I didn't miss anything from the presentation, but I wonder if you have been working in nick points, these geomorphic features that you find in streams that can be also hot spot of mixing.
Yeah, I think nick points would be sort of like one of those reach scale features, you know, that happens at the scale of basically like hundreds of meters to kilometer, right? And so then you would tend to, because of that profile, you would tend to get probably, I mean, I'd have to think about it, but you would probably get more of upwelling of groundwater down of the nick point. Do you see that at a field site where you work or something like that? Yeah. And then that would be superimposed with a lot of these smaller scale exchange processes that are also happening. And like Daniel mentioned, you know, maybe there are places where it's just all broader groundwater upwelling or downwelling, and then other places where hypereic exchange becomes more important again because you have more neutral conditions.
So yeah, you can go.
I just have one quick
Oh yeah.
Here, coming back to the streams.
Yeah.
Here in France in the 70s, they remodeled lots of streams so that they went really straight to help farmers, make it easier for them to farm the land. And it was an ecological disaster because of the lack of diversity in the habitats. That
happens a lot.
From your talk, I now understand that it was probably also impacted a lot the exchanges between the boundaries and the rivers. And I was wondering whether the relation between the shape, not of the bed, but also of the river itself,
Yes.
and the exchanges have been studied systematically.
It's true that these meanders are another building block for hypereic exchange and are really important because they also end up creating, as you have flow around meanders over time with these restoration projects, they will naturally sort of form pools and riffles that are sort of like giant bed forms too. So you get both types of exchange, the flow kind of laterally as you describe, but then also flow through new features that are forming in some ways because of the flow around the meanders, and it's all connected, I think, when we make our streams very simple to improve flow conveyance, like get the flood downstream, let it go. We had probably hundreds of years of doing that to our rivers, including something near and dear to my heart, taking wood out of streams because it's debris, it's ugly, it causes flooding, but we lose that connectivity and that complexity that drives so much of the ecological heterogeneity and the chemical heterogeneity that we care about. So maybe that's a bigger picture than what you were going for. But if you want to learn more about the meander flow, Daniel's got a poster out there too.
Okay, so I would like to thank the speaker again